We begin with the FASTQ files, which contain the raw unprocessed reads. The reads undergo pre-processing, typically involving quality control filtration and trimming, as well as logistics facilitating steps like splitting the data up for parallel processing on multi-threaded computing clusters. Once the reads have been pre-processed, they are aligned against a reference genome. Unaligned reads are typically trimmed and submitted for a second alignment attempt. This process produces a SAM file of raw alignments. If the pipeline was parallelized, it will produce multiple SAM files, which are then combined into a single SAM file. From this SAM file of raw alignments, valid, unique HiC pairs are identified, and general metrics on the data are reported. From the HiC pairs, we can infer the frequency of loci association, which is then used to draw biological conclusions. Note that you don't have to run all these steps manually. Several end-to-end bioinformatics pipelines exist which carry out all three steps. Anyway, now that we've done the overview, let's go into a bit more detail. I'll start by explaining what FASTQ files are. FASTQ files store DNA sequence data along with the quality scores for each nucleotide. The format could theoretically work for amino acid sequences, but personally, I've never seen it used for that before, so in the context of this video series, I'll only talk about FASTQ as a file format for DNA sequences. In FASTQ files, each DNA entry represents a single sequence of DNA, known as a read, and consists of exactly 4 lines. The first line contains the read ID. The second line contains the ACGT nucleotide sequence. The third line is irrelevant and often left empty. The fourth line contains the quality scores of the nucleotide sequence. The FASTQ file format doesn't have a way to store additional information, like secondary structure or molecular modifications, but that kind of information doesn't exist in the NGS data anyway. Single-end sequencing is when only one end of each DNA fragment is sequenced, while paired-end sequencing is when both ends are sequenced. Paired-end sequencing produces results with higher mappability than single-end sequencing, but costs a little bit more. Some techniques or analysis methods explicitly require paired-end sequencing in order for it to work. HiC is one of those techniques. HiC sequencing data is thus always paired-end. Paired-end sequencing typically produces one pair of FASTQ files per sample per flow cell, one file containing the forward reads, and one file containing the reversed reads. Multiple samples sequenced on the same flow cell will produce multiple pairs of files. A sample sequenced on multiple flow cells will also produce multiple pairs of files. Very occasionally, both the forward and reverse reads are merged into a single FASTQ file. General best practice is, for each sample, to have all the forward reads in one FASTQ file and to have all the reverse reads in another FASTQ file, resulting in one pair of FASTQ files per sample. Most HiC pipelines require the data be input in this manner. FASTQ files for a HiC pipeline can come directly from a sequencing experiment, from an online repository, or both. When it comes to the pre-alignment pre-processing, there are 4 common steps, not all of which will take place. There's adapter and primer trimming, quality control trimming, restriction enzyme site trimming, and the combining or splitting up of files for logistical reasons. Since both the input and outputs of all these steps are FASTQ files, they can technically be performed in any order, although reliable reporting metrics may require a particular order. Once the pre-processing is finished, the FASTQ files can be aligned against the reference genome for that organism. To be more precise, they are aligned against the index of the reference genome, which needs to be generated. However, once a reference genome index has been generated, it can be used to align any number of FASTQ data files and does not need to be generated again each time. Sometimes, the unaligned reads are aligned a second time after being trimmed. This is usually only done for HiC, which uses restriction enzymes to fragment the DNA, and in pipelines for such DNA in which restriction site trimming was not carried out during pre-processing. The mapping process produces a SAM file. So what is a SAM file? SAM stands for Sequence Alignment Map, and this is the file format used to store alignment results. Some alignment programs produce BAM files instead, but BAM files are just compressed SAM files. They take up less hard disk space on your computer, but they contain identical data, organized in the same way as their uncompressed counterparts. I will explain SAM files in detail, but you can also refer to the official SAM file specifications document linked in the description below. The SAM file format can be described as a table with header comments. That is to say, a SAM file consists of two parts, a comments section and a data table. The comments section consists of lines which begin with an @ symbol and are typically used to record metadata, such as the details of the alignment procedure which was used to derive the alignment data. The data table contains the alignment data itself in a TSV format. TSV stands for tab separated values, so in raw data form, different rows of the table are separated by new line characters, while the cells of each row are separated by tab characters. Each row of the TSV section corresponds to the alignment results for a single read. The first column contains the read ID. The second column contains information on how the alignment went, and this information is stored as a number which uses the bit flag system. The third and fourth columns contain the chromosomal coordinates of the best mapping result. The fifth column contains the mapping quality score. The sixth column contains more information on how the alignment went in a cigar format. The seventh and eighth columns contain the chromosomal coordinates of the next read in the data. The ninth column contains the fragment length of the sequencing fragment, which is only applicable to paired end sequencing, while the 10th and 11th columns contain the DNA sequence and quality scores of the read respectively. These are not always identical to that of the corresponding entry in the original FASTQ file. Some SAM files have additional columns with additional information, depending on which program generated them. The most important information lies in the third and fourth columns, which contains the chromosomal coordinates of the alignment. Now, in conventional experiments, read pairs are aligned together and considered to be two ends of the same locus. In HiC, this is not the case, and the reads are aligned separately. Okay, since we now know what SAM files are, we can talk about post-alignment processing. There are a lot of artifacts in HiC data, and the purpose of post-alignment processing is to remove those artifacts and give us an idea of how efficient the experimental process was. Technically, the first sub-process of this step is to recombine files if samples were split up for parallel processing. If and once that is done, we can move on to the actual bioinformatics steps. Firstly, the read pairs are separated into candidate pairs, singletons, and other. Candidate pairs are read pairs where both reads align uniquely. That is to say, each read only aligns to one location in the genome. Singletons are read pairs where only one read aligns uniquely and the other read does not align anywhere. Some pipelines also regard read pairs as singletons if one read aligns uniquely and the other read aligns to multiple locations. Secondly, the candidate pairs undergo validation. Invalid pairs are excluded, leaving behind only the valid pairs. Thirdly, deduplication is performed to remove PCR duplicates. Both the valid pairs and the singletons undergo deduplication. Deduplication isn't always performed though, for reasons we'll touch on later. Finally, the unique valid pairs are transformed and normalized into loci interaction or association data. Some methods use singletons during the normalization process. Not all steps have to be performed in that order. Recombining split-up files must take place before deduplication, but before then, all steps can be parallelized. Most HiC pipeline implementations don't bother with parallelization of the post-alignment steps though, so files split up for the alignment step are usually recombined immediately. Identifying candidate pairs must be performed before validating them. The screening out of singletons and multi-aligned reads informs us of the bioinformatics limitations of the alignment step, while the screening out of invalid candidate pairs tells us of how well the HiC wetlab procedure went. Removing the bioinformatic artifacts first leaves us with just the wetlab artifacts, hence the order. Deduplication can actually take place before the identification of and validation of candidate pairs, but not if those steps are parallelized. Therefore, HiC pipelines usually deduplicate their reads as late in the pipeline as possible, either because they have implemented parallelization for previous steps, or because they might do so in the future. Also, deduplication might not be viable for data which used restriction enzymes for the fragmentation step, since it will be possible for chimeric fragments to form independently of each other, which start and end on the same nucleotide. As mentioned previously, some HiC data normalization methods make use of the singleton alignments, but this is not always the case.